Okay, I'm already presented. You can see my screen, I suppose. Yeah. So good evening. So welcome to week 7 of Bioengineering course, which is an interface with biology and medicine. So myself Gaurav, and I'll be just initially giving a recap of the week 7 lectures which Professor Sanjeeva has taught, and then we'll move on to question and answer session of previous year assignments, and we will try and solve them and discuss. Yeah, so what has Sir majorly talked about in terms of week 7 lectures? So he initially talked about amino acids and later about proteins, like how exactly what all types of amino acid are present and uh, how they make up into a specific protein and how different structures of proteins are present, primary, secondary, tertiary, quaternary, and how do they function, and how the structure of a protein exactly happens, and how can we determine those structures, how can we analyze the functions of proteins, etc. All those were discussed. And then in proteomics field, exactly those functions and uh, from a gene. Initially, genome was given a lot of importance. Then, after understanding the genome, people understood that understanding protein is also equivalently important. Then, people tried to understand proteomics a bit and tried to analyze how proteins function and how they are important to us. And lecture 33 and 34 majorly dealt with different techniques to understand protein, like what all techniques generally used for understanding protein extraction, protein separation, or uh, uh, during 33 and 34th lecture. And in the 35th lecture, sir gave an overview of bioinformatics. So in that bioinformatics also, like how do we align two sequences if there is a similar sequence or if there is a different sequence, etc. All those were uh, like all those were explained, like how can we do that in a in silico way. So why studying proteins is important because from DNA transcription happens to RNA, from RNA proteins. So proteins are actually the one which perform different functions in an individual. So understanding proteins is important. How they are made up of? So they are made up of amino acids. This is the basic structure of amino acid, which has the amino group at one terminal and carboxyl group at other terminal, like other side, and hydrogen bond is there, which so this carbon is called as alpha carbon as there are four bonds attached to it and the side chain. So this specifically changes as per the specific amino acid. So if you see one side has amino terminal, one side has carboxyl terminal, one has H, and the side chain keeps on changing. The yellow one, if you can see here. So accordingly, the amino acids are named, and each amino acid has been labeled with a three-letter word, which is for glycine it is GLY, alanine it is ALA, and similarly they are also coded with single-letter word as well. So accordingly, you need to so you should know all of this three-letter code and single-letter code. So it's not exactly like for glycine it is G, alanine it is for A. For example, there are few which has different one-letter word. For phenylalanine it is F and not P. Tryptophan it is W and not T because they have been already used for tyrosine and proline. So that's the reason they have a different one-letter word. And accordingly, the side chain of it is different for each amino acid. That's how different amino acids comes into picture. And according to these side chain present, they are also divided into like aromatic amino acids or non-polar amino acid or polar acid like hydrophobic or hydrophilic, all different types of. So we can have different classifications of amino acid depending upon the side chain. And there are some amino acids which has sulfur in it, for example, methionine and cysteine. So this is one cysteine which has sulfide bond in them, like this methionine and cysteine. And these are few examples of polar or hydrophilic amino acids, and these are examples of hydrophobic amino acids. Similarly, so that depends upon the side chain itself, as I said, and we can also so depending upon polarity, these are the difference. We can also divide them depending upon their charge. So it can be either acidic or basic, indicating that either positively charged or negatively charged. So if the side chain has carboxyl group, so this has a negative charge. So this comes under aspartic acid and glutamic acid comes under acidic amino acids. And if the amino acids have a positive charge, NH2 plus amino group and the side chains, they come under basic amino acids. So which for example lysine, arginine and histidine. Similarly, these comes under aromatic amino acid and they can be also called as hydrophobic characters. That's how it decreases and the hydrophilic character increases, but they can be apart. So as they have aromatic ring in their side chain, they come under aromatic amino acid. And if you generally take an absorption spectra of a protein, in general any protein, and this at 260 to 280, if you see at this particular wavelength, the absorption is maximum. That maximum absorption occurs because of the presence of tyrosine and tryptophan. So if your protein has a major amount of if there are 4 to 5, for example, if a protein is around to 100 amino acids, for example, and out of that 100 amino acid, maybe 10% of it or maybe 15% of it has tyrosine or tryptophan, then this specific can you hear me? Hello, can you hear me? Yes, Srishti, can you hear me? I think it is showing me certain sign that, yeah, can you hear me? Yeah, I think you can hear me. Okay, yeah, there was some, because of the phone call, it happened that the audio was not working. But you did hear, right, what I talked about amino acid and stuff? Yeah. Okay. So this is how. So this specific spectra is observed only when uh, if a major amount of tyrosine and tryptophan are present in a protein. Similarly, what are peptide bonds? So peptide bonds are nothing but a bond between two amino acids. So if this is one amino acid methionine, this is tyrosine. The bond between them, which happens between a carboxyl group and an amino group, that bond specifically is called as peptide bond, which actually joins the two amino acids. And that's the reason, because as this is peptide bond, that's the reason a chain of amino acid is called as a polypeptide. And how does the band actually occur? So there is a removal of water and only accordingly then NH and CO comes together to form an peptide bond. So that's how a peptide bond is formed. Similarly, the rotation of it is flexible. So this specific bond is there, and this actually helps the rotation of this specific amino acid differently with the help of, so this psi angle and phi angle can change depending upon uh, the strength of peptide bond and depending upon the specific side chain amino acids. And but there is a specific angle at which they are stable, and they cannot be stable in each and every angle. So those specific angles are given by Raman spectra. So we'll be talking about that as well. And there are few amino acids which can or there are specific amino acids which are non-chiral in nature, or they are having chirality, which, so what that chirality means, so that indicates that they are non-superimposable to each other. Non-superimposable in the sense, they, if you keep one amino acid to another amino acid, they will be not be the same. Like for example, you, if these two hands are together, so they are not same, right? So here one thumb and this is one thumb. So so they are not getting superimposed with each other. So just like two hands are non-superimposable mirror images of each other, amino acid molecules are also non-superimposable due to their chiral alpha carbon. So that happens because of the chiral alpha carbon. So that helps in the rotation of this. And because of that chiral alpha carbon, there are different types of rotation of. So basically, that rotation of D isomer or L isomer depends upon the polarity of light. So if a polarized light moves towards right, whatever direction, accordingly, it is called as either a D isomer or L isomer. So dextro-rotatory or levo-rotatory. So like clockwise or anti-clockwise. So whatever specific amino acid rotates at what specific angle, either dextro or levo-rotatory. So dextro are always having their side chain at the left side, and uh, levo-rotatory always has their side chain at the right side. So accordingly, those specific amino acid rotate this polarized light in that specific direction, and hence they are named as D isomer or L isomer. And each amino acid behaves differently in different specific pH. For example, in terms of glycine, so if it is in an acidic medium, when you add that specific amino acid in an in order to understand like what might be the charge of a specific amino acid at a specific pH solution, so if the solution pH is acidic in nature, then what is the charge of glycine? So glycine is generally positively charged in an acidic medium, so it will move towards the negative terminal. Cathode is negative terminal, anode is positive terminal. So and so if you add this specific glycine in, which is in the acidic medium at the center of the uh, electrophoresis paper, then it will move towards uh, cathode, that is towards a negative charge, because glycine is positively charged when it is in the acidic medium. So this indicates that your amino acid is positively charged in acidic medium as it is moving towards cathode. And the detection of this amino acid can happen with the help of ninhydrin when it is sprayed on ninhydrin. And when that specific if you increase the pH a bit from acidic pH to neutral in the range of neutral pH, then hydrogen is lost. So basically, this initially it is positively charged at acidic pH, then that hydrogen is lost and it becomes, so there is one more, so this hydrogen is lost and there is negative sign which is created. So this is plus sign as well as minus sign, making it neutral. So this neutral pH is called as PI value. So at this specific pH, glycine has this specific form of glycine which has plus sign as well as negative sign. So this specific form of amino acids are called as deuterionic forms, or else they can be also called as isoelectric point. The point at which, so what is isoelectric point? It is a point at which amino acid has neutral charge. So at this specific point for glycine, they have a neutral charge, and they can they will not either go towards cathode or on anode side, they will be in the center. But when you add, uh, make it bit more neutral and add much more base to the solution, so you can see that there is one more loss of hydrogen ions, and this plus sign also gets disappeared, and there is only negative charge. And here the amino acid is negatively charged because you have increased the pH a bit. And at that specific pH, then if you want to test the charge of glycine, you can again add it on a paper electrophoresis, and you can see that now glycine will move towards anode because it is negatively charged. And this is the point at which it will move towards anode, and this is the point at which it will move towards cathode, and this is the point at which it will neither move towards cathode or neither move towards anode. So that's how people have done different experiments and understood how amino acids behave at different pH, and people now know at what specific pK it will move towards cathode, at what specific pK it will move towards anode, and at what specific pH it will neither move to any of the direction and be a neutral in charge or a deuterionic form of it. So for each amino acid, there are different values of this according to their side chain. So that is what we can understand that amino acid behave differently at different pH. And what is the functions of proteins in general? So they have enzyme catalysis, they have transport and storage. They can be responsible for our immunity. They can be responsible for growth and differentiation, also as a mechanical strength. So there are different functions of protein, and they play a key role in structural as well as certain functional roles. So they help in cell signaling, they help in transportation of certain molecules from one part to another part, they help in catalysis, they help in movement, like muscle strength and everything. They have a specific structure, and depending upon the structure, their function is determined, and they also help in regulation of our body. So how is exactly protein formed? So initially now we have studied about amino acids here. So how exactly a protein is formed? So proteins are made up of amino acids. Then each so after making these peptide bonds, so that linear protein is called as primary structure of the protein. Then amino acid sequence. So this is the amino acid sequence for a protein. So what is the significance of that? So essentially, it helps us to tell us the mechanism of action of a specific protein. It determines the 3D structure of protein. So whatever is the sequence, that will also help us to understand the 3D structure of protein, as well as if there is an alteration, how it can lead to certain abnormality, and the sequence will also tell us about the evolutionary history of the protein. If that same protein is found in some other organism also, and to recently, like how the basically evolution has happened of that specific protein. If you know the sequence, and if you also know the sequence of earlier organism, you can just correlate and understand how evolution has happened with the help of proteins. And there are different ways of representing the protein. Either it can represent in ball and stick model or ribbon diagram. So this is how they look. And what is the secondary structure? So if you know the sequence of amino acids, so that basically folds itself in a different forms, and one of the form is alpha helix. And how does that folding happen? So that interaction of folding happens between the first residue with the fourth residue. So if this is the first amino acids, 1, 2, 3, 4, 5. So this fourth amino acid, so this is first, this is second, this is third, and this is fourth. The fourth amino acid which forms the hydrogen bond with the fifth amino acid. So this specific carboxyl group interacts with the amino group of the fourth amino acid. So after this is 1, 2, 3, and 4. So after this amino acid, you need to count 1, 2, 3, and 4. So that fourth amino acid, the amino group of that interacts with each other, and it twists a bit, and it kind of forms an helical structure. Similarly, the second one interacts with the fifth amino acid, and that's how the interaction happened to form an alpha helix, and these bonds are hydrogen bonds. And in terms of beta pleated structure, so there are two amino acid sequences. There is one amino acid sequence and the primary structure of it, and second primary structure of it. So these two primary structures interact with each other here, like the amino group and the carboxyl group, amino group and the carboxyl group. So these two interact with each other to form a parallel sheet or an anti-parallel sheet. It can be either in the parallel form or an anti-parallel form. Parallel in the sense both are in the same direction, anti-parallel in the sense both are in the opposite direction. So like N terminal to C terminal, N terminal to C terminal. Either in the same direction, then it is parallel. If it is opposite, it is anti-parallel. And what is a tertiary structure? When this alpha and beta structures come together, they form a tertiary structure. So this is an example of myoglobin or hemoglobin, wherein an alpha structure as well as loops here. You can see certain loop here. So they all come together and form a complete uh, structure of a protein. They are called as tertiary structure. And how they are studied? They are studied by X-ray diffraction method. So there is a crystallogram which is created, and using this crystallogram and using certain softwares, people try to understand the structure of a protein. And in terms of quaternary structure, so it happens when one specific amino acid sequence has one structure and other amino acid sequence. There are two different chains coming together, or three different chains coming together, or four different chains coming together. Uh, so each of them has their own tertiary structure, and when those all of these each tertiary structures come together and form one protein, then it is called as a quaternary structure. So here there are four identical polypeptides. So there are four. So one specific structure, tertiary structure of one polypeptide like this, identically four are there, and when they come together, then transerythrin is formed. So this has four identical polypeptides. So that's how it is. Similarly, hemoglobin is also kind of that structure where it requires four identical subunits. So these are the different levels of protein structure: primary, secondary, tertiary, and quaternary. So this was an animation of the same. If you have looked at the videos, uh, so each amino acid coming together with the help of a peptide bond. And as I was talking, so this specific rotation and forming the secondary or tertiary structure, so there are only at specific angle it is stable, and it is not stable at each and every angle. So this phi angle and psi angle, so if there are 5 angle degree and psi angle degrees here, if you see those, so those are actually stable at these specific regions only. If you see here, so beta sheets are stable in this dark red regions, and if you see the right dark one, and alpha helix are present in. So if there is an alpha helical structure, these are the angles which actually makes a specific protein stable, and for beta sheet, these are the angles which actually makes a protein structure stable. So only these are the specific angles which actually helps a protein to have a stable structure, and it cannot form. So this specific rotation has certain limitation, and limitation in the sense they can freely rotate, but they cannot be stable, and the amino acid can only be maintained and stable only at these specific angles. So this the same thing, like it was just an animation of it in the lectures. So basically, in beta sheets, so there are two specific amino acid sequences, and they interact. And this is going towards this direction, N terminal to C terminal. And if there is an interaction between these two by hydrogen bonds, and then it is called as anti-parallel beta sheets. And this is parallel beta. So there are specific phi and psi angles required for its stability. And if these two come together, like alpha helical and beta sheets, it forms a tertiary structure. So these are beta sheets, secondary structure, and when they come together with each other, it forms a tertiary structure. And hydrophilic groups are generally outside, and hydrophobic groups are inside, making it soluble protein. So this was it in terms of first lecture of week 7, and coming going ahead with week second lecture of week 7. So sir initially talked about genome sequencing project. Why? Because he just wanted to give a recap of importance of genome project, which actually helped all other to understand that when you know genome, you should equally know understand proteome as well. And people then, after these genome projects were successful, people understood that we need to understand proteome as well. And people then, from 2003 or in that 2000 era, people went ahead and tried to uh, make more scientific research related to proteome project. And when they know the genome completely. So for that, so proteome was started in the later era, like completely focused in the later era than that of genomics. So what is proteomics? Basically, it is a study of proteins and the properties to provide an integrated view of cellular process. So it can be, so all these techniques are a part of proteomics itself, localizing the protein, where exactly proteins are produced, where it is getting transferred from one cell to another cell, or from one organelle to another organelle, all those. And even protein-protein interaction, how a protein interacts with some other protein or with some other. There is also protein DNA interaction as well. So how does that exactly happen? How does a protein exactly function? How protein gets modified from one form to another form during post-translation modification? All everything is a part of proteomic study. Whatever happens to protein in general is a part of proteomic study. And so what are the facts and perspective of it? So proteomics aims to study the extent of expression of the protein itself, how translation modification happens, how enzymatic regulation happens, like how activation and inactivation of a protein. So specifically, if a reaction needs to take place, an enzyme will come and it will convert from form of a protein to another form. But it should convert only to certain level, right? It should not continuously being active and continuously forming that specific form, inactive form to active form. So that specific enzyme needs to be regulated. It should have certain active nature at whatever whenever it is required, and it should be inactive as well whenever it is not required. So all those regulation, how does it exactly happens? Similarly, what kind of protein-protein interaction, the localization, structure, function exactly, all those are understood in proteomics field. So to give an overview how a protein is formed, so from DNA, RNA will be formed, and that in that RNA exons are the one which are coding form, and introns are non-coding ones. So introns are removed with the help of alternate splicing, and exons are formed. So this specific gene here you can see it actually form two different kinds of proteins by joining different exons. So here exon 1, 2, and 4 was joined, and here 1, 3, and 4 was joined. And because of this specific joining, different kinds of proteins were made. So each gene can make different number of proteins. So maybe if I give you a broad overview, for example, if there are thousands of pre-mRNAs, maybe there are lakhs of proteins which can be made from those thousands of pre-mRNAs. And different types of modifications happen. If there is phosphate group attached, it's called as phosphorylation. If glucose or carbohydrate is attached, it's called as glycosylation. If a methyl group is attached, it's called methylation during the post-translation modification, or if it is hydroxyl group, it's called as hydroxylation. That's how different protein modification happen. Uh, this was an animation of Human Genome Project, that different countries were involved in it, and people tried to understand the whole genome. A part of each, like people tried to read each part of the chromosome and make the whole study possible. And it was started in the year around 1996, and by 2001-3, it was 2001 around 80% of it was completed, and 80 to 90% of it, and later by 2003, the Human Genome Project was completed. Basically, Human Genome Project indicates that the whole sequence of the whole human body, DNA sequence. But during that specific project, there were many major hurdles, and people tried to overcome those hurdles, and people tried to find things. So, what all things were found was there are around 30,000 protein coding genes, and so there are 3 billion base pairs if you see here, but out of this only 13,000 proteins are coding genes, and all other are non-coding one. And hundreds of human genes may be resulted from horizontal transfer from bacteria and mutation. So this is all these were found after the Human Genome Project was successful. Mutation rates in males are twice as high as in females, and recombination rates tend to be higher in distal regions of the chromosomes. So distal regions in the sense these regions. So they understood that in these specific regions, recombinations are greater rate as compared to that of the central regions. So these are conserved. If you see, most of the human beings might be having these sequences very much conserved, and whereas these sequence might be different in most of the individuals. And there have been more than 1.4 million single nucleotide polymorphism identified on the human genome. So these many number of single nucleotide polymorphism indicates, for example, there are 100 nucleotides. In that 100 nucleotide, there is only one nucleotide which is different, and all other 100 nucleotides are similar in most of the individuals. So that one change is called a single nucleotide polymorphism. So this is just an animation of the same which we just talked about. And so so that was about genome. Then people understood that it is also important to study proteome. Then people came up with proteomic study, wherein all this in the year from 1975, people developed different techniques, and by then people understood that it is very important to understand all of this and learn, make use of all of these techniques continuously. Different algorithms were developed accordingly, and 2D electrophoresis, mass spec, that is uh, LCMS, like mass spec based using chromatography as well, and there are chip based approaches as well. So basically, if you want to analyze a protein, there are four different things you should know, like understand like how exactly protein is extracted, how exactly protein is separated, then how exactly you can identify the protein. So in terms of separation, there are gel-based method and there are gel-free methods as well. And in terms of identifying, there are mass spec-based methods. There are uh, mm, bioinformatics based methods. So there are different methods to identify a protein. Similarly, for characterization, there is microarray, and there are different techniques available to do all of this. So these are the major. So this is the same thing. What are different kinds of techniques are available? 2D, dyes, microarrays, etc. So how does it perform? So in terms of 2D gel electrophoresis, so there is a uh, Sir has also given a demo of how exactly to uh, perform this 2D gel electrophoresis as well as differential gel electrophoresis, which is DIGE. And so how exactly it is performed has been given a demo as well as we have understood it. In terms of 2D electrophoresis, initially isoelectric focusing is done wherein an IG strip is taken, which has different pH values here, and according to the pH, uh, an electric current is given, and proteins migrate in a specific direction according to uh, their pH. And if a protein achieves its goes towards its isoelectric point, then it stops migrating. So that's what we studied in our amino acid part of this lecture. And so accordingly, at that specific pH, it will stop moving, and it will have no effect of the electric current as it is the net charge of the protein becomes zero. So it doesn't move any of the direction, and that's how we can separate the protein depending upon their pi values, which is isoelectric point. And then this same strip has been kept in the SDS page gel, and that same strip then allows the separation of those amino acids depending upon their molecular weight. So this separation is depending upon their pi, and this separation is depending upon their molecular weight. So that's the reason it is called as two-dimensional electrophoresis. This is first dimension and this is second dimension. And in terms of dyes, so there is an internal standard uh, which is a protein sample, and they are dyed with and there are this is, for example, this is the sample, and this is the standard 1 and 2, and this is another sample, and they are dyed with different dyes, and then then the same thing is performed, isoelectric focusing and then SJS page. So if as we know the uh, bands, the intensity of these bands corresponds with the sample 1 and sample 2. So this indicates that in the sample 1, these are the few things, like if you can see, as this is the standard one, you all know what all kinds of proteins are present in this, and you have uh, stained it with certain specific dye. And when you mix all of this and uh, make a pool of it and then do an isolectric focusing and SDS page, then you can see that there will be different colors, different. So this you will be running in different gels, and you will see according to the dye which you have provided and different colors of like, after visualization, you can see that what kind of proteins might be present in your sample by comparing with the internal standard. So that's how digest perform. So these are gel-based proteomics, and there are mass spec-based proteomics as well, wherein in your sample itself an ion source is injected and the samples get ionized, and when it's getting ionized, so depending upon their mass and charge, the mass analyzer depending upon the mass by charge ratio, so that is charge and m is mass, and depending upon their mass by charge ratio, it will so there is an ion detector which will try and detect them, and if in your system itself it will uh, form different according to the mass by charge ratio, it will give different relative abundance, and then data is processed and people try to analyze the sample accordingly. So there are different types of ion sources. If you see, uh, like for example, MALDI, wherein an ion source is injected on the uh, target plate where your sample is kept, and that sample gets ionized. And once it gets ionized, so depending upon their mass and charge, there is a flight tube where, according to the mass and charge of this particular ions, they get separated, and that is one method which is called as MALDI-TOF. And there is one more method where ESI method, wherein ion source is there in a needle and it is specifically given certain voltage and it is pushed, which actually creates different ions, and that specific ion is now taken up by the um, sample orifice, and sample orifice in the sense the uh, nozzle basically where it is getting inserted. So accordingly, so in terms of this analysis, there are different types of analyzers available which can analyze the mass of an ion as well as charge of an ion. So it depends upon time of flight. There is quadrupole, there is ion trap, there is ion cyclotron, there is orbit trap, there is magnetic sector. So different types of analyzers are available. Majorly time of flight, TOF is used, or quadrupole is used, and orbit trap is used. So these are the major analyzers which are generally used, but people, depending upon the study, people also use these other uh, analyzers as well. So how does that exactly function? So there are two of which are actually explained in the lecture, which is time of flight and quadrupole. So what happens in time of flight? So there is a long tube itself which helps the ions to travel a bit. So once the sample is injected with the laser which ionizes the sample, once the sample is ionized, depending upon their mass and charge, it gets separated slowly. So the smaller ones get reflected towards the detector initially, and the larger ones get detected later, depending upon their mass size. And that is in terms of the time of flight. And in terms of quadrupole, so there are four poles available which actually makes the so there are different mode of actions. You can either do it in scanning mode or refractive mode. So in that specific radio, basically a radio frequency mode, in that radio frequency mode, Depending upon the mass by charge ratio, they can pass through this specific quadrupole after it is getting ionized. And in terms of scanning mode, what happens is that then we will have to give certain uh, range of size or range of mass, and accordingly it will uh, allow only those specific ions to pass through this quadrupole and go towards the detector, and all others are ignored. There are different modes available in terms of quadrupole. So these are different analyzers, TOF, Q, ion trap, etc. And these are ion sources. How exactly ion source are given? So either it can be MALDI form or it can be in the ESI form. So they are conjugated with different and typical different MS configurations are available. People either go with MALDI TOF or ESI QTOF or ESI ion trap or MALDI ion trap, etc. So depending upon type of analyzer you want and type of ion source you have, uh, different ways of analyzing the mass by charge ratio of a protein. So generally, how the if you want to study protein-protein interaction, so if there is an host specific protein and you have engineered the strain and it is expressing the protein, all this we have studied in terms of cloning, if you all remember. And once that is expressed, you will do the purification of it and run the gel. And after running the gel, that purified, you will purify the protein. That purified protein is now added on a specific plate where it can interact and we can study the interaction of it. So generally, proteins have different functions. It can be used for signal transduction. It can be used in DNA replication. It is used in metabolism. It is used in cell growth, etc. Then it has different functions. So in terms of their interaction also, in terms of interacting with nucleic acid, proteins, lipids, or small molecules, whatever interaction is happening for. So that interaction can be transient or that can it can be permanent. And this interaction can be of whatever kind of bonds it is forming, it can be of any form. It can be either electrostatic interaction, it can be hydrophobic interaction, it can be hydrogen, or else it can make certain hydrogen bonds and then interact. It can be different ways to perform its functions. So in order to understand that, there is one technique called as microarray technique, wherein initially there is a glass plate where proteins are printed basically on it, and if your protein of interest is interacting with that specific protein which you have plated, then it will interact and it will show. Then that interaction is basically understand is scanned by this micro array scanner, and if it is interacting, then it will give the. So that is basically attached to a die, and if the interaction happens, then the die will illuminate. So if you see, so this each and every dot is having certain specific proteins which you have printed, and your interest of protein you are just adding on to it. So your interest of protein you will add on to this, and if there is an interaction happening with whatever specific proteins, then it will form certain it will interact with that specific protein. It will the dye will illuminate, and that is analyzed by microarray scanner. And if you see, there are only few specific wells which are shining, indicating that it is only interacting with those specific proteins and not all the proteins. Then there is label-free detection techniques as well, wherein there is no label. So here there is a label which is attached, and like green color label or red colored label which is attached. So accordingly, protein is getting eliminated. Whereas here there is no label attached as such. So there are also techniques wherein it is purely depending upon antigen antibody interaction. If there is a complex form, then there will be change in reflection angle, and if there is no complex form, then the angle will be same. So that's how you can understand whether interaction is happening or not happening, depending upon the reflection angle. So there are different applications of it. If you're taking the blood sample, analyzing it, or using or taking a tissue biopsy, and then doing protein analysis and microarray. So different applications are there. You can either use a blood sample or a tissue biopsy and try to understand what kind of proteins are present in it. All of this can be done using different techniques. So what all techniques are there? We'll be studying that. And if you see, this have been discussed in many of the lectures after genome, whatever transcript form that is transcriptome, and whatever proteins form that comes under proteome, and when these interact with each other and form different metabolites, that is formed as metabolome. So these were the conclusions in the after the second lecture. Then from the third and fourth lectures, majorly talked about different techniques. So we'll take a break of like 5 minutes or so, and then we'll go ahead with this uh, third lecture brief overview. So let's take a break of like 5 minutes, and we'll be back. Okay. मी येतो तोपर्यंत निघून जातो वाटतं. अरे एवढं खेच नको नाही. तो हालेल एवढं खेच पण. Okay, we'll start it again. So till now we talked about the initial two lectures of the week 7, and then we'll move on to the techniques which are available for understanding the proteins. So one of the major technique which is generally used to separate proteins is electrophoresis technique. Uh, Generally for DNA we use H, which is called as agarose gel electrophoresis, but in terms of protein, I have on the audio, right? Yeah. Okay, so for proteins, it is polylitramide gel electrophoresis. It can be either with the help of SDS, so making it making it rod shaped and similar charged, or else we can it can be the native as well without using SDS. So what does electrophoresis? The basic principle is migration of charged proteins in an electric field. So in an electric field, they will be migrated depending upon their charge. So basically that happens with the help of molecular weight majorly. So so we have already discussed about two-dimensional electrophoresis wherein initially the suppression happens with the help of isoelectric focusing depending upon the pi value. So wherever the protein has a specific pi, it stops the migration over there because at that specific pH it is having zero charge. So that's how the suppression is happening in terms of isoelectric focusing. And the same strip is used for suppression of uh, specific protein depending upon their molecular weight. So this is the second dimension, this is the first dimension. So now the protein suppression happens on the basis of uh, molecular weight. So that is SDS electrophoresis. So this is just talking about how exactly that strip looks, and these strips are generally more stable and durable because gel is prepared with plastic backing that that ensures pH gradient is fixed in place. So there is plastic as well, and in that plastic there are pH gradients fixed in the place. So it is comparatively much more stable and durable as well. And how exactly we perform? So first equilibration is with all of these specific components, which saturates IG strips with SDS and reducing agents. And the second, so this is the equilibration steps for this specific isoelectric, the first dimension itself. So initially you need to equilibrate this specific membrane itself before adding your sample. So that equilibration happens, the first happens with this, all of these components, and second happens with these components, and each one have their own role. For example, here urea will denaturate the protein, and similarly DDT will be there, and here, which will denaturate the protein. And here, in the second equilibration, iodoacetamide is also added, which actually prevents protein reoxidation, and I'll call it residual DTT to minimize vertical streaking. So vertical streaking in the sense that it will not spread away as such. And in terms of the second one, SDS page. So how does this SDS page gel is made? So there was a demo in your lectures. You can also look at that demo as well, how exactly in the lab SDS page gel is formed. So each specific component of that SDS page has certain role. Acrylamide has a role for making the matrix, bisacrylamide for cross-linking those acrylamide and forming the cross-linking so that it forms spores by making the cross-links, and the protein can be separated. And for cross-linking and for the polymerization, there is an initiator which is called as APS. And SDS is the one which gives the negative charge to the protein, and it also makes protein rod shaped and as well as gives negative charge to the protein, and beta mercaptoethanol, which is BME, which breaks the disulfide bond. So if there are if your protein of interest is made by two specific separate amino acids like polypeptide chains by which are joined by disulfide bonds, then it will be separated and only single. So that if you are adding beta mercaptanol, then it will be separated. If you are not adding, then it won't be separated. So depending upon the study, you can accordingly use it. So after running the gel, so there are different dyes available to visualize those proteins. Most majorly it is chromatin brilliant blue which is generally used, but there are other dyes also available which can be used for visualizing the protein. So similarly in terms of DNA, if you might be knowing, we generally use ETBR to visualize or cyber grain to visualize. Similar to that concept, so in order to visualize the protein, we have different dyes. So more elaborative expression of the same thing, like how uh, isoelectric focusing happens. Like if there is a separation between, so there is a gradient of pH, and the molecules have, if there are net positive charge, it will be towards the acidic range. If it is no net charge, it will be in the between. If it is going towards 7 and having negative charge a bit, it will move on and move till 7. And in terms of, so this have separation happens in the first dimension, and the second dimension it majorly happens with the help of molecular weight or size, you can say. Yeah. So if there are two conditions like this, condition A and condition B, you need to solubilize the protein, you need to run them as per the isoelectric point from minus, if protein is positively charged, it will stay here. If it is protein is negatively charged, it will go on moving. And once it gets zero charge, then it will stop over there. So that's how the separation happens. And after that separation, you will be just adding that same strip onto the LTS page, and the proteins will get separated depending upon the molecular weight, and you can stain the gel using different dyes, and then you can visualize them. So accordingly, you can understand that at what condition, what kind of specific proteins were there. So at condition A, what kind of proteins were there, and in condition B, what kind of proteins were there. So basically, such experiments can be performed, and 2D will help you to understand how exactly it's functioning. So in terms of diets, if you see, there are sample and there is a control, or you can say internal standard, and accordingly those will be labeled differently. A sample will be labeled differently, and your internal standard will be labeled differently, as well as your samples will be labeled differently. And if you pool them together and run it in the same way, dies way, then you can from your image analysis. Uh, so basically, in this image analysis, what you will do is you will specifically try to visualize one specific dye at a time, and accordingly, you can understand uh, from the whole picture and background your how your so basically you will compare with the internal standard itself and understand how your proteins were are getting separated. So this talks about how your internal standard should be made. So basically, you'll be taking few microliters from all your samples and grouping them together, and that will be called as a pooled sample or the internal standard. So these were certain gel-based techniques. So there are some other techniques also in order for separation of proteins. So one of the technique is gel filtration chromatography. So how does that happen? So there is a gel beads itself. So these are certain beads which have pores in them. So if you see the larger particles here move comparatively a little faster because they cannot enter the pores of this specific uh, gel beads, and whereas you can see the smaller proteins which are smaller in size can enter these pores, and because they are entering this course, they move within the beads, and they move comparatively a little slower compared to those of larger sized proteins. So they can move whatever spaces are present between the beads. So they can basically move with these spaces between the beads and move a little faster, which are larger molecules compared to that of smaller molecules. So that's how separation happens in terms of gel filtration chromatography. And in terms of ion exchange chromatography, so depending upon the charge of, so this is not similar to that of isoelectric focusing. So here also there is a charge to an protein, and it stops when so this separation is on the basis of isoelectric point. So it will continue to go until and unless it gains zero charge. So where there will be no effect of the electric charge. Whereas in terms of isoelectric ion exchange chromatography, you need charge. You cannot make it zero. You need the charge. So you will be accordingly using the buffers of that specific pH so that it maintains that specific charge. So proteins are separated based on the charge difference itself. So proteins will overall negative charges will interact with this positive charge or vice versa. So it can be either anion exchange chromatography or cation exchange chromatography. So it can be either anion like negatively charged exchange chromatography or positively charged exchange chromatography. So accordingly, there are certain beads available for it. So for there is carboxymethyl group and there is a dimethyl aminoethyl group. So these are certain beads, like beads are of cellulose, and there is a group which is attached to it, which is negatively charged. So this is used for anion exchange chromatography, and these are positively charged, and this is used for cation exchange chromatography. So accordingly, for example, if beads are negatively charged, then negative negative will repel each other and negative amino acids will move a little faster, whereas positive will interact with these specific beads. And once you will do the elution of it, and then only the interaction uh, will disappear and the positively charged amino acid then get separated. So initially all the negatively charged amino acids get separated and then later positively charged amino acid will get separated. So that's how ion exchange chromatography works. So if there are if you are adding beads which are negatively charged, then you will adding a protein solution which there are certain proteins which positive charge and certain proteins with negative charge. For example, then accordingly, if it is negative, then positively charged protein will interact with it, and whereas negatively charged protein will get eluted first, and then when you do the elution of it at that time, positively charged proteins will get eluted. That's how you can achieve separation. And similarly, in terms of affinity chromatography, so this depends upon the how much affinity a specific amino acid or a protein is having with those beads. So in terms of his tag, so his tag has certain affinity towards nickel. So these specific beads are having nickel to it. So there is load on nickel 2 plus column. So these beads has nickel attached to it. And if a protein of your interest is having his tag or histidine patch, 6x histidine patch or 10x histidine patch, so that will interact with this specific beads which has nickel and with the help of imidazole, so you can wash it. So basically, imidazole has much more interaction with nickel as compared to that of histidine. So once you add this imidazole with higher concentration, what will happen is that imidazole will try and bind with this nickel and it will elute the histidine tagged protein. And that's how you can separate your histidine tagged proteins. This is called as affinity based chromatography. So there are different types of chromatography methods available for separation of proteins. So these were the one ion exchange as well as gel filtration or affinity based chromatography. So these were the techniques which were explained in your fourth lecture, and then in the fifth lecture of week seven, Sir majorly talked about how bioinformatics can be used. So he talked about NCBI website where in the database you can select different stuff. So you can also search for papers in this NCBI. So for example, one of the examples sir gave in terms of protein identification. So basically you need to select protein here, write the protein name. If you write the protein name, you can actually get this IDA and all of when it was updated, when it was identified, what is the code of the protein, how many amino acids are there, what is the genetic code of that specific protein, how many nucleotides are present, like basically how many base pairs, from which organism it is. All these details can be obtained just by uh, writing the proteins here, and if you search it, then that specific protein will have all those details. Then in terms of alignments, as I was also talking about whether if you want to understand the evolutionary or if two proteins are similar or not, if you take it from one organism, if you take it from another organism, and if you know the sequence of it, and if you want to understand whether this the sequence of these two proteins are similar or if they are different. So you can do blast of it accordingly. So blast indicates basic local alignment search tool, and so one subunit of which has 142 amino acid, you can just align that protein with some other protein. So basically, BLAST will help you in alignment of two nucleotide sequence, one nucleotide and one protein sequence, protein and nucleotide sequence, or two protein sequence. It can do all of these stuffs, and it will help you to understand uh, how significant, how similar they are, or how different they are. And if there is a change in one single amino acid or one single nucleotide, everything can be understood using this BLAST. And if you want to understand multiple sequence at one specific time, so this will allow you to do two sequences at a time or three sequences at a time. But if you want to do multiple alignment at one go itself, then you can use another software which is called as Cluster Omega, and that will help you to understand uh, if you want to understand everything in one go itself. So that was it in terms of understanding or just giving a recap of all the five lectures. Yes, I hope you are getting it. And so that is what Sir talked about in terms of the first lecture of, sorry, all the five lectures in terms of week 7. And we'll move on to the previous year assignment questions. So we'll again take a break of around 5 minutes and we'll go ahead with the questions. If you have any doubts till then, maybe we can discuss. Yes, yes. If you have no doubts at the time, and when we discuss questions, if certain doubts arise during the question understanding, we can accordingly discuss at that time. So we'll be back in like 5 minutes, and we'll discuss the previous year assignment questions.
हाँ मैंने उनको बोला बात करने के लिए। 7। 7 बजे। 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 मुझे नहीं पता। ठीक है। हाँ, क्योंकि हाँ, मतलब उसका चेहरा तो नहीं है क्लस्टर को, तो मैंने मॉर्निंग लगाया था उसका प्राइवेट पर। ठीक है। ओके। हाँ, ठीक है।
Okay, then we'll start the lecture again with the previous year assignment questions. So, do you have any doubts before that? You can write it in the chat box if you want. Yes. Should we go ahead? Am I audible? Okay, yeah. So should we go ahead now? You can write it in chat box if you want. So I want you to answer certain questions. So if you want, you can speak up or you can write it in the chat box. I'll keep on switching this. Maybe we can do it this way. Can you read the question even now? Uh, Yeah, you can do it this way. So what you can do is you can write your answer here in the chat box and you can discuss the questions. Okay. So tell me, so there is a team of researchers who has purified two novel proteins. So there's one novel protein A and one novel protein B, which has a tag of GFP and has a tag of RFP. That is green fluorescent protein, red fluorescent protein. And two novel proteins A and B, which have a length of th 300 amino acid and 400 amino acids. Okay. And to study the intercellular localization of protein, they were expressed in a cancer cell line fused with two fluorescent uh, tag proteins. So these are two fluorescent tag proteins, GFP and RFP, which has a molecular weight of 27 and 25 kilodaltons. So this has a molecular weight of 27 kilodaltons and this has 25 kilodaltons. What are the approximate molecular weight of this fusion protein? So fusion in the sense protein A plus GFP, protein B plus RFP. So that is the fusion. Given the average molecular weight of an amino acid and water are 110 kilodalton. So we have given a total number of amino acid of each protein, protein A and protein B, and the average molecular weight of an amino acid is 110. So accordingly, you can use these values to understand the molecular weight of protein A and protein B. You already know the molecular weight of uh, GFP and RFP, which is 27 and 25. So can you calculate what will be the total molecular weight of protein? Yes. Can you calculate this? Which one should be the answer? First, yes. And for protein B, okay. So how did you calculate it? If you can explain. So how did you come up with 60? Yeah, you're correct. So that's how you can calculate. So you know how to calculate. Great. So the answer is the first one, 60,000 and 69,000. Yeah. So the answer is first one, 68,000 and 68,000. Okay. So now the protein is so as the protein A is tagged with GFP and protein B is tagged with RFP, at which wavelength would you visualize them under confocal microscope? So this will be okay. So this will be known only if you know at what specific wavelength green is visualized and what specific wavelength red is visualized. So this is something question you need to learn it and then only you will be able to answer it. So if you know, so basically if you might have known WebGear, that specific wavelength. So red is generally visualized at very high wavelength and violet is visualized at very low wavelength. So similarly with GYOR, so R will be at majorly at the visible range at the after 700 around 700 nanometer, and green will be somewhere between, and whereas violet will be around 200 250 something. So if you know, if you want to visualize green and red, so it should be so if red should be approximately at the 700, so this should be the answer, and green will be obviously here. Yeah, so ideally B should be the answer. So if you are not aware of it, maybe you can just try and find out where yellow is visualized, green is visualized, red is visualized. All this wavelength, approximate wavelength you should know so that you can answer this question. And the answer is B itself. Okay, so upon visualization of both proteins using high resolution microscope, so these two proteins were visualized and they were found to be in peroxisome. So this is a peroxisomal structure, and it was found that both of these proteins are present in the peroxisome, and they resembled the image depicted below. So this is how the image it was, RFP filter. So when only green was visualized, this is how the image was, and when only R was visualized, this is what it was seen. And when both of them were together visualized, so this is how the visualization was. So please explain what does this yellow color signifies. So people have tagged with red fluorescent protein and green fluorescent protein. Then how does this yellow color is coming into picture? So when the expression of only green and red fluorescent proteins were being monitored, then how does yellow is coming into picture? So there are different options. The yellow color signifies the debris that were produced during the sample preparation. The yellow color signifies the co-localization of both proteins together, or yellow color comes from a new chemical compound as a fusion of proteins A and B reacts with each other in peroxisomes, or both A and B. What do you feel? Yes? What do you feel? The answer should be no idea. Then if you can write in the comment box, you can guess any of it if you maybe option B or C. So the correct option is B. Basically, there is co-localization of it. So there are two proteins are expressing together here. So when green and red is mixed together, so you will obviously see yellow color. So there is no interaction as such because fusion proteins of A and B is not the thing. The fusion is between uh, protein A and GFP and protein B and RFP. There is no fusion between protein A and protein B. So there is no fusion like this. The fusion is between A and GFP and B and RFP. And whatever yellow color is visualized, that is because of green and red are mixed together. So both are getting expressed together. So basically, if you express green and red together, the expression will be yellow in color, right? And if you only see green, it will be green. If you only see red, it will be red. But if you see both of them together, it will be visualized as yellow color. So that's what is happening here. So both proteins were visualized. So here, if you see, green was also present in all of these structures, and red was also present. So if you see both of them together, it will appear as yellow. So the answer is B. Yeah. So the next question is, so this is a schematic representation of a peptide. So so chemical bonding between amino acid and the fundamental building blocks of protein. Which of the following statement of peptide bond is correct? So where is peptide bond? The peptide bond is between carboxyl group and amino group. So this is a peptide bond, and this is a peptide bond. So you need to understand the correct statement out of it. So what is the correct statement? The 3D folding of the protein happens because of interaction between R groups of amino acid with carboxyl group in peptide bonds. So the Peptide bonds is not happening with the interaction with the R group. The peptide bond is between carboxyl group and amino group. So this sentence is probably not correct. You need to find the correct statement. So the peptide bonds are very flexible and can rotate freely in all directions. So as we already studied that they are flexible, they cannot rotate in all the direction because there are certain limitations of their direction as we studied with the Raman spectra which we were dealing with during our lecture. So even this is not the correct statement. The formation of peptide bonds occurs through dehydration synthesis. The water molecule goes away, yes. So wherein the amino group, so this amino group of one amino acid reacts with the carboxyl group of another amino acid to liberate a water molecule. Yeah, this statement feels correct. So hopefully this is right. But let's see the fourth option as well. So peptide bonds are formed through hydrolysis, breaking the connection between the carboxyl group and amino group of adjacent amino acids. So we are talking about the lysis. So there is nothing like lysis. Basically, there is a formation and not nothing like breaking it down. So it's not this is not the correct one. So answer should be C. You're getting it right? Yes. Okay. So now, so as I was talking in terms of page, it can be either native page or SDS page. So using size exclusion chromatography. So this is gel filtration chromatography itself. You have purified an 100 kilodalton protein. So there is a protein which is 100 kilodalton. Okay. To check the quality of the purified protein, so you want to check the quality of it. So when you perform page in the presence of SDS and beta mercaptoethanol, so in beta mercaptoethanol it will split it. If there are two peptide bonds present, it will break that because we are adding beta mercaptanol and as well as SDS. So it yielded two different bands. So these were the two different bands, each having a molecular weight of 20 kilodalton and one was 30 kilodalton. However, when you run a native page, when there is no breaking, so there is no SJS page, there is no beta mercaptanol here, so it will be uh, together and it will run. So there is no breaking as such. And when they did that, they actually found an 100 kilodalton band. So to ensure reproducibility, the purified protein was loaded in triplicates. So this is just for reproducibility. This purified protein is loaded in triplicates. Every time in both cases, a known protein marker was also run. So this is a known protein marker knowing the molecular weight to track the molecular weight. So below is the representation of it. So what is the best explanation of this scenario? The protein is has three subunits. One has 100, one having 100 kilodalton, one having 30, and last one has 20. So if it would have three subunits, then there should be a band at 100 kilodalton also in this, along with 100, 30, and 20. But there is no 100, so it is not a three subunit protein. The protein is either homotetramer of 20 kilodalton or homotetramer of 20 kilodalton subunits. So 20 into 4, if you see tetramer, it becomes 80 and it doesn't become 100. So even this option is not correct as such because tetramer indicates 4, right? So 20 into 4 becomes 80. So but the size which we are getting is 100. So this is not the correct answer. This page was faulty, hence multiple wrong bands. So basically it is done in triplicate, so it cannot be faulty and all of them are giving the same result, so it's not the answer. The protein is homodimer of two heterodimers, 20 kilodalton and 30 kilodalton. So 20 plus 20 homodimer of each of two heterodimers, that is 20 and 30. So 20 plus 20 becomes 40 and 30 plus 30 becomes 60 and 60 plus 40 will give you 100. So this is the answer probably. Yes. So two 20s will give you 40, two 30s. So homodimer of both of them, homodimer of 20, homodimer of 30, both joined together will form a 100 kilodalton protein. The answer is fourth one. Okay, so a synthetic peptide contains four negatively charged and eight positively charged amino acids at pH 6.1. Okay, so at that specific pH, there is a peptide bond, which overall peptide in the sense there are different amino acids, out of which four amino acids are negatively charged and 8 amino acids are positively charged at that specific pH. Okay. So when the peptides are treated with alkali solution, the pH is now increased. Positively charged group becomes deprotonated. If there is deprotonation, it will become negatively charged. So this positively charged proteins become deprotonated. So proton is eliminated. So they will become negatively charged or they will become zero charge. Whatever. Let's see. So at pH 8.9, what is the net charge on the peptide? What do you think? So now pH has increased and these positively charged amino acids have become uh, deprotonated. So there is no positive charge left. So they might be neutral now. What will be the direction of movement? Towards cathode or towards anode? Cathode is negatively charged, anode is positively charged. So overall proteins, what will be the charge of that overall protein and what direction it will move? If the alkali treated peptides are now placed in an electric field. So now this after treatment they are placed in an electric field, what in which direction it will move and what will be the net charge. So zero movement of the peptide will not happen. Minus 8 movement of peptide will happen towards the anode. 12 plus 12 a moment of peptide will be towards anode and minus 4 moment of peptide will be towards cathode. What do you think? Yes, so this 8 amino acids positively charged become deprotonated. So positively charged. Okay. So it is say oh sorry. When the peptide bonds are treated with alkali solution, so four positively charged, not all of them. Only four positively charged groups have become deprotonated. All other four are still positive and 4 are negative, and 4 have become deprotonated at this specific pH. So what is the net charge now? Option A, yes, you are right, because 4 have become deprotonated, so it is having 4 positive and 4 negative. 4 positive, 4 negative becomes 0, and the movement will not happen. You are right. Okay, the arrangement of many polypeptide chains or subunits into a functional multi-subunit complex defines a protein quaternary structure that we know. Which of the following statement about quaternary structure is accurate? Okay, so when does a quaternary structure form? When different tertiary structures come together, like each specific two peptides come together and form a quaternary structure. So let's see which one is correct, which one is more accurate. So the beta chain direction in a protein's primary structure is only a factor that determines its quaternary structure. Not exactly. So regardless of size or purpose, all proteins have their quaternary structure. No, all proteins doesn't have. There are if there is an amino acid with only single peptide, that can only finally have tertiary structure. It cannot have quaternary. Quaternary structure involves the non-covalent interaction and spatial arrangement of multiple polypeptide chains, possibly. Quaternary structure is only relevant when it comes to enzymes and has a role in structural transport proteins. Which one do you think? You can make a guess if you want. Option C, maybe. Yes, you are right. Congrats. So basically, we need to eliminate the uh, option. So here, by explaining itself, I've eliminated all of these. So this might be the possible answer. So that's how you can solve these questions. Okay. So a differential DIGE was performed with two distinct tissue samples, and normal and one cancerous. Then a combination of both normal and cancerous have been combined with all three dyes in that order. So whatever order here. So the normal tissue was compared with has a dye called Psi2, cancerous has Psi3. and pooled sample has sci-fi. Okay, they were mixed together, done isolatory focusing, gel electrophoresis, etc., gel scanning, and when they were scanning at different specific filters of those specific dyes, this is what how the visualization was. Okay, so now the samples were combined, put on the gel according to cyanide dyes. The gel image was scanned at three distinct wavelengths. Okay, so select the right statement if this is the pattern. Okay, so if you see, these are few things which are getting matched from cancerous, and these things are from a normal tissue. Let's see what the gel N plus C always has fewer spots than gel N plus C together. So if these are together and these always have smaller, fewer spots. Do you think so? The gel N plus C gel excludes the probability of batch to batch variation and is considered to be a good reference for future data analysis. The protein in gel N and gel C are entirely different. They are not entirely different. If you see, there are a few specific proteins like which are similar also. So if you see this specific line is kind of similar. So this is not the right answer for sure. So it can be both AB or only A or only B. What do you think? So this is also not a correct answer. If you see N plus C always have fewer spots than gel N and C together. It's not that always it will have fewer. It might have more also. So because it is a pooled sample, it has these proteins as well as these proteins all together. And this, so what is the option you feel? So basically, initially when you take these samples and you store it and you only use this as a standard, so this basically helps in batch to batch variation, understanding if you take a different batch of this sample and then run it, you need to only compare with this. If it is present in this, then only you will consider those bands. And if it is not present in this, you will not consider those bands. So basically it helps in understanding the batch to batch variation and acts as a good reference. So that's the reason a pooled sample is always necessary. The answer is B. So now you have identified an unknown protein which has a molecular weight of 27 kilodalton from MS-based proteomics. So you have done mass spectrometry and non-proteomics and understood that the size is 23 kilodalton and it plays a crucial role in cervical cancer. You have further sequenced the protein to get amino acid sequence. Now you wish to study the conserved regions of the protein. So what bioinformatics tool you will use to understand about such crucial information about the protein? So protein has a cervical cancer load and you want to, now you know the sequence of it and you want to find the conserved regions. So it was easy, we already studied about it and you need to use bioinformatics tool. Okay. So run a blast to get a similar sequence from different organisms, perform multiple sequence alignment to look for conserved regions. Yes, this is what the answer should be, but let's see the other options as well if there are more accurate options. Analyze the protein tertiary structure. So this is the primary structure, right? So it is telling us to analyze the tertiary structure and perform protein-protein interaction. So it is very complicated and you don't have to do all of this stuff to understand the conserved regions as such. So because here the question is not about understanding how it interacts, how exactly it has what kind of structure it has. If that was the question, then B might be the answer. Here you want to find the conserved regions. So probably A should be the answer. But let's see, predicting protein secondary structure using computational algorithms. So they are telling us to predict secondary structure first. Will immediately give you information about the conserved regions of a protein. So understanding only that one specific protein and understanding its structure will not tell you about the conserved regions. Or option A and C. So it's not that way. So this will just tell you about the secondary structure and it will not tell you about the conserved region. So this is an incorrect option. Only BLAST will help us to understand conserved regions in the sense with all those different generations, that specific part of the protein is similar in most of the organisms, different organisms. So that's what conserved region talks about. So answer should be A. So you have identified a novel drug that can bind to an oncoprotein, that is cancer protein, named SRSF3. How will you visualize the protein drug interaction? So now we are talking about protein drug interactions and gain more idea about binding site and its interacting amino acids. So how will you find that? We'll perform a protein purification followed by LCMS, perform drug toxicity assay such as MTT. So that will not tell you about the interaction basically. You want to understand protein drug interaction. Search protein database for its structure and perform blast to know the drug binding site, possibly, or perform a molecular docking followed by visualization using bioinformatics tools such as PyMol. This could be the possible answer, or the fourth one could be the possible answer. Which one do you feel? So this will basically purify it and separate it. That will not tell you about the interaction. The question is, you need to visualize protein drug interaction. So depending upon the question, so the question might be changed and option will also be different for that specific, like the answer can be different. So depending upon the question, what is the answer? What do you feel? Yes, option D, yes, you are right. Basically, this will just tell you about the conserved regions by doing blast, or it will by taking its structure and basically will right. So blast will basically help you to understand. Basically, it will align different proteins. It will not tell you about visual. It will not tell you about which is the part which is interacting. So Pymol basically helps you. Molecular docking will help you. So this is the answer. Yeah. So that's it. Maybe we can meet the next lecture, during which will be the last lecture, the week 8 lecture. And if you have more doubts related to anything, we can discuss regarding that, or else we can close the meeting. Yes, anything else you want to discuss? Yes, Srishti, no doubts. Great. Uh, where are you from, by the way? What are you doing? BTech, MTech, any course specific? And you are from which institute? Oh, BTech first year. BTech in biotechnology? Nice. Great. So if you still get any doubts while solving assignments, etc., you can email me. I hope you all have my email IDs, like the TA's email ID. So you can email me if you want any doubts we can clear at that time as well. Thank you. If you have any doubts, we can discuss, or else we can discuss.